Good afternoon. Good afternoon and welcome to today's edition of Science Sundays. I'm John Beacom, I'm the chair of the Science Sundays Committee, and I'm from physics and astronomy, but I'm representing a committee that's from across all of the arts and sciences, including natural and mathematical sciences, social and behavioral sciences, and arts and humanities. So, the starting point of science is curiosity. And the second part is research. But the third part is sharing what you learned. And we share that not only with our peers through our publications and our students through our classes, but we share it with the public. Because science unshared is like science undiscovered. It doesn't do you any good, it doesn't do the world any good if you know something and you don't tell anybody. So here we are, we're telling you. And I'd like to introduce my colleague, Professor Meg Daly, who's going to introduce today's speaker.
Thanks, John. It's my pleasure today to introduce my colleague, Andrea Grottoli, distinguished professor in the School of Earth Sciences. Dr. Grottoli began her career with a bachelor's from McGill in her native Canada, followed by a PhD at the University of Houston, postdoctoral fellowship at UC Irvine, and a brief stint as an assistant professor at the University of Pennsylvania. Through those experiences, Dr. Grottoli developed an innovative research program combining geochemistry, marine and environmental science, and coral physiology to understand how the health and success of reef animals changes under different environmental conditions. In the 20 years since Dr. Grottoli has been at Ohio State, her integrative approach to working at the intersection of climate, ecosystem, and biology has proven vital for interpreting and predicting the health of coral reefs. Dr. Grottoli has been a leader in addressing these globally important questions, serving, among other roles, as the lead on a research coordination network on coral bleaching, as a Fulbright Fellow in France, as a Fellow of the American Association for the Advancement of Science, and as the President of the International Coral Reef Society. We're fortunate to have her here today to broach this question, can we save coral reefs? Please join me in welcoming Andrea Grottoli.
Thanks for having me. It's a pleasure to be here today. And I'm happy to talk to you about can we save coral reefs. Before I get started, how many of you have ever been swimming in the ocean? Almost everybody. How many people have had a chance to open their eyes underwater, either snorkeling or something like that in the ocean? Okay. Any scuba divers? Wow, welcome. And how many people have seen coral reefs with their own eyes? Excellent. So a lot of people have seen coral reefs here. Well, hopefully when we talk about coral reefs today, then it's not quite as foreign. For those of you who haven't had a chance, hopefully I'll give you enough here to give you a good sense about coral reefs. A healthy coral reef looks like this, right? We have plating corals and bubbly, these mounding corals that make this whole tapestry of coral. And coral are the building blocks of coral reefs. They make the structure that all the organisms associated with reefs need. You also have this whole layer of fish associated with coral reefs. And inside all the cracks, there are a whole bunch of invertebrates. There are crabs and lobsters and worms and all kinds of fun things. This is another example of a healthy coral reef in the Line Islands. That's in the middle of the Pacific. And in this example, we can also see some of the larger predatory fish, these silvery jackfish of some kind. There's some sharks in there. We also have dolphins and whales that can associate with coral reefs at different times. And so these are very complex, multi-structured, three-dimensional environments. But I want to take you on a virtual dive, and we're going to test my technological skills here in doing this. This is in Indonesia. This is through the Catlin Seaview Survey. And this is showing you what it would look like if you were diving on the reef yourself. And you can see all the different kinds of corals. There are fingering ones and bouldering ones. There are fish over top. What you can't see is in the cracks and underneath there are sponges, worms, crabs, lobsters, starfish, every kind of critter you can imagine. And it looks like this is a spectacular example of a healthy coral reef. Unfortunately, coral reefs are highly degraded throughout the world. And this is the result of a multitude of things, including ocean warming, ocean acidification, pollution, overfishing, and disease. Warming is one of the single largest global threats to coral reefs. Because we keep burning fossil fuels to power our cars, to warm our homes, to cool our homes, to generate energy, this is increasing the amount of greenhouse gas in the atmosphere. Greenhouse gas warms the planet, it warms the atmosphere, and it warms the ocean. The oceans are increasingly warm, and corals are living closer and closer to their thermal thresholds. And what happens is when corals are exposed to temperatures above that threshold, it causes stress, and it causes bleaching, which can lead to mass mortality. So in this example from American Samoa, we can see the reef was healthy in December of 2014. There was a large heat stress event which caused bleaching, and the corals turned white. They're very unhappy. They're not dead yet, but they're not happy. And then when the heat event lasts long enough, they can't recover and they eventually die. So in this last image from August 2015, the corals are all dead. And we see this happening more frequently, and the events being more severe, and the events being longer as time progresses. And so while pollution is a real threat, overfishing is a real threat. It destabilizes the ecosystem. Ocean acidification, these are all real threats to coral reefs. On a global scale, temperature is the single largest threat to coral reefs, and I've spent a lot of my research studying the effects of temperature on coral reefs and what we can do to save them. Since 2000, coral cover has decreased by 12% globally. In some places, it's decreased a lot more. In other places, like in Indonesia, the coral cover there has remained relatively stable. But in most places, it's been going down, and in some places, catastrophically. For example, in Florida, coral cover has decreased by 90% since the 70s. So we're going to take a tour here of photos. This is Carey's Fourth Reef in 1975 in Florida, where we have this large cover of elkhorn coral or Acropora palmata. They covered everything. Very healthy, very bright, vibrant. By 1985, they were mostly dead because of disease. The source of that disease is still debated, but they got a lot of disease, and there was mass mortality. You can't tell in this picture. There were some that still survived at this point, but since 1985, that particular site has continued to decline. That species has continued to disappear throughout Florida. And by the time we get to 2014, it has, not only is it dead, but everything that's dead hasn't been repopulated, and so it's eroded to rubble. Recovering a reef from this state is very difficult. And we are increasingly seeing rubble-filled, completely devastated, eroded reefs around the world. And in Florida is a particularly egregious example. In 2023, there are parts of Florida that still had pretty healthy reefs. But in 2023, there was a mass bleaching event that was catastrophic throughout the Florida Keys. The video on the left is at Chica Rocks, one of the places in Florida that had some of the healthiest reefs. And you can see that the corals are all white. So a healthy coral is brownish or greenish, and white means it's bleached and it's not happy. And all of these corals are bleached. These are all mountain corals. You can even see the sea fans in the foreground here. Those are all dead and dying as well. This reef actually recovered pretty well. But everywhere where there were the branching corals, like the one on the right here, this is the Acropora palmata again. Acropora palmata and Acropora cervicornis, the two keystone species, historically the most abundant ones throughout the Florida Keys, have been dwindling over time, and the ones that were left, 70% died during this bleaching event. And this particular species is now effectively extinct in the Florida Keys. There are a few colonies that remain. They're too far apart to sexually reproduce effectively. So this species can only be saved in Florida through human intervention. Coral reefs are in a crisis. And at the core of this is warming. At our current rate of warming, we can see here on the top graph, the red line, these are sea surface temperatures that have been measured since 1950 that are grouped with projections going forward at the high emission scenario. This is a scenario where we continue to burn fossil fuels at the rate that we are now, and by the end of this century, the oceans will warm by about 4 degrees Celsius relative to where they were mid-last century. If we mitigate climate change and we go with this low emission scenario, warming will still be about 2 to 2.5 degrees Celsius. The low emission scenario is the Paris Climate Accord agreement scenario, one which we are not abiding by, and most countries that have abided by are struggling to meet, and globally we are following the red line. So a colleague of mine did some modeling exercises based on what we know about the extent of corals and coral biology to see how they would fare going forward. Under the low emission scenario, if coral can adapt, the percentage of coral that will exist relative to today at the end of this century is about 30%. This is the blue line. This is the best case scenario. With the track we're on, even if corals can adapt, the expectation is that coral will be completely gone by 2050. Now, this is a modeling scenario. It doesn't take into account every nuance or every place on the planet, but it does show the degree of stress and crisis that we're under. My group has done research where we've grown corals for 2 years under the low emission scenario, and we find that about 50% survive. So we got a better outcome than this model. We got about 50% survival. So there may be more resilience than we thought, certainly in the low emission scenario framework. At the high emission scenario, the path we're on, all bets are off. It's really hard to know what will happen. All indications is that it's probably really bad. So why does this matter? Why would we care about coral reefs? Well, coral reefs, it turns out, have huge value for humans. Humans' well-being is linked to coral reefs. We depend on coral reefs for food, pharmaceuticals, spiritual and cultural values. Coral reefs are hubs of marine biodiversity. Almost 1/3 of all marine species are associated with coral reefs, even though they occupy less than 0.1% of the ocean. They are equivalent to the rainforests on land in water, right? They have that level of species diversity and impact. They are important for coastal protection. Coral reefs strongly reduce wave energy and wave height. They mitigate flooding, and this saves money and lives. And finally, coral reefs play a pivotal role for jobs and the economy. Coral tourism and fisheries provide significant income and revenue. Globally, it is estimated that coral reefs have an annual economic value of about $10 trillion, US dollars. In Florida, it's about 20 billion annually, just Florida. So can we save coral reefs? We know that they are important for humans and for the reefs themselves. And what's not on my list there is they're also beautiful. They're incredibly beautiful. They're amazing. They're places where you go diving and it's a Zen moment, right? You feel all that around you. Reefs make sounds. There's like clicking sounds from shrimp and fish. There are bubble sounds. It's this beautiful place. So even though they have value and they're economically valuable and they protect coastlines, they're also amazingly beautiful. So can we save them? Sure. We need to mitigate climate change and local stressors. It's a pretty tall order and something that many people are working on. And it's something that, globally requires a lot of effort and global effort. So that's taking time. We can also conserve what exists and restore what has been lost. And a lot of work is being done here. This is work we can do now while we're working on the climate change problem. But the rate of change is so fast that restoration efforts can't keep up. And the corals that you restore are also still subject to the same stressors that were there before, which are climate and pollution and overfishing. And so all of that has to be addressed at the same time. And what we're discovering is that we need some technology, or we can use technology in some cases, to help this along, to help restoration work better, last longer, and be more effective. So here are some examples of restored reefs. On the left is a degraded reef from September of 2015 in Indonesia, and on the right is that reef in 2019. I don't know if that reef is still there today, if they've had a bleaching event and everything has died, But in that window, it was successful. And what I'll point out, it's not just the coral that are there, the coral are here, is that you also see all the other organisms, all the fish associated with reef are there. Reef fish need corals for hiding, mating, food, sleeping, protection from storms, protection from predators. When fish are on reefs like this, they'll swim around. If they detect some kind of threat, they sink into the corals. If there's no coral, they can't live there. Here's another one from Indonesia, before and afters. So this restoration can be effective if those corals can survive, if they can reproduce, if they can produce new offspring to keep the reef going. And that is proving to be a very large challenge. So one of the things we can do to help this is use technology. And I have invented a device that helps coral eat more, grow more, and die less. And I'm going to tell you about this device. But to do that, I want to share with you just why that works, what would make me want to do this in the first place. How do coral make a living? What is a coral? And how can technology help them? And how can we restore them using technology? So first, we got to understand what a coral is. This is one kind of coral. This is a mounding coral on the left here. This is Orbicella fabulata. It's probably about 200 years old. These big mounding corals lay down skeleton in layers, just like trees have tree rings. Corals have bands. They lay their skeleton down in bands. And the living tissue is only a couple centimeters on the very top. So the brown you see there is this veneer of tissue over the top, the living part of the animal. And then it has skeleton underneath that's dead, but it keeps growing on top of it. And that's what builds the structure of a coral reef. And corals are two things. They're an animal host first, but they're symbiotic with algae. They have single-celled algae in their cells, and that's what makes them brown or green or whatever color they are the symbiotic algae. And so because they have these two things, they actually get food two ways. That's what mixotrophic means. They have two ways. They are animal first. If we zoom in here, we can see it's made up of all these little polyps, and the polyps have tentacles and a mouth, and they can capture stuff with those tentacles. And the stuff they like to capture are things like zooplankton. This is a zooplankton here. These little guys are less than a millimeter long, and coral can capture them with their tentacles, put them in their mouth. And they're nutritionally dense. These have vitamins and minerals. They're complex organic sources of food. But inside the cells, if we zoom in a little more, of the animal are the endosymbiotic algae, or these single-celled algae. And they have 10 to the 6, or 1 million of these cells per centimeter squared of coral tissue. So like the area of the end of my pointer here is about 1 centimeter squared. That would have 1 million algal cells in it. And they photosynthesize far in excess of their own metabolic needs and translocate that excess sugar to the coral host, giving the coral host fuel. In return, the coral host eats zooplankton, gets complex molecules and nutrients and vitamins that it can share with the endosymbiont. And so this is how that partnership works. Healthy corals can get 100% of their metabolic needs met through photosynthesis alone. Metabolic means just like fuel for keeping the engine running, for breathing, growing, sleeping, that kind of stuff. I don't think they sleep, but unfortunately, when corals are exposed to temperatures of as little as 1 to 2 degrees Celsius above their normal thermal maximum for as little as 10 days, it causes that symbiosis to break down. The coral get rid of their endosymbiotic algae. And what used to be brown, like you see in the top right here, brown healthy coral reef, turns white. They get rid of their endosymbiotic algae, and we see through the clear gelatinous tissue to the white skeleton underneath, hence the term bleaching. This is a very stressful condition for coral. They're not dead yet in this case, in this image, but they're very stressed out, and they're not getting enough photosynthetic sugar, so they're in a bit of a starvation state, and even more dependent on the zooplankton that they eat. Under these conditions, if the waters don't cool off soon enough, they will start getting really sick, and eventually they start dying. But there are always some coral that are more resilient than others. So if we look at this picture down here, everything is white, and then in the top left of this picture, there's this one brown coral. It's doing fine. It's more resilient. There are always some tougher ones out there. tougher under today's conditions, right? The scenario that's putting pressure on coral reefs now, this coral is able to tolerate it a lot better. So what makes some coral more resilient than others? And work I've done in my lab and in other groups as well have pointed to several things that make coral resilient. I'm going to selfishly only cite my own papers, but know that other people have done this work as well. But I'm probably the only person, my team is the only team on the planet that's doing measuring all of these things and thinking about all of these things together at once. So one of them is they can change the species of algae they associate with. So it's not just one species of algae in the corals, but there are a suite. Some can switch them. Not all coral species can. Some can only have one, and that's it. And if it doesn't work, too bad. Others can switch them out. And so shuffling their species of algae is one trait associated with coral that are more resilient. The microbiome. In humans, we know the gut microbiome is really important for human health, and we think it's also true for corals. So there's a whole bunch of people studying the microbiome of corals. What my team has found is that coral either with a stable microbiome or that can shift their microbiome to be more diverse tend to be more resilient. Corals with high energy reserves, particularly fat. When coral are bleached, they're not getting sugar from photosynthesis, so they're a little calorie deprived. If they have fat reserves, they can use that fat reserve to sustain them. Corals that are fatter do better under stress. They're more likely to recover. They're less likely to die of bleaching. And then feeding. Corals that can feed a lot naturally on zooplankton or that can increase their feeding in response to stress do better, because now they can access that other way they get food. They're less dependent on photosynthesis, and they're better able to cope with that stress event. So I'm going to focus a little bit more on the feeding part, and that's where I've spent a lot of time in the last decade of my work, because it turns out feeding is a critical source of nutrition. And I can give you a couple of examples. We did a study in Hawaii where we measured how much of the metabolic demand corals could get just from photosynthesis alone and from feeding alone. And we did this with 3 species, Porites compressa, Porites lobata, and Montipora capitata. These are species I've worked with a lot. They're very common in Hawaii. And what we can see in the graph here is the y-axis shows percent of metabolic demand they're able to meet. And if the dashed line shows 100%, so meeting all of their respiratory needs in a given day. For Porites compressa, the dark line bar here is the proportion of that demand met from photosynthesis alone when they're healthy, and it more than meets 100%. They're doing great, and that's true for all 3 species. And then there's a little top up here of what they get from eating zooplankton. We know that even though in this scenario, when they're healthy, they don't need the zooplankton to keep themselves fueled, but we do know they need it to grow because they need those complex molecules, nutrients, vitamins, things like that to build cells. You can't build cells on sugar, but you can build cells if you eat zooplankton. So in a healthy coral, they all can get, do fine with photosynthesis, little top up from feeding, but when they're bleached, and they don't have their symbiotic algae, then they can't meet 100% of demand from photosynthesis. Porites compressa here is way below 100%. The feeding puts it over the top. So coral feeding enables bleached corals to meet metabolic demand, and this was true for all 3 species. Especially interesting is Montipora capitata. Notice when it's healthy, it has very little feeding, this red bar here, but when it's bleached, it's gigantic. It's almost 100%. They ramp up feeding. They don't even care that they're bleached. They can just switch to their other way of feeding for a while till conditions get better. And then when conditions are better, it grows back up its algae, it tunes down feeding and goes back to being photosynthetic, right? It is very flexible in how it gets that food energy. And the other ones aren't nearly as flexible, but thank God they feed, right? Because it helps them get over that hump. So feeding enables bleached corals to meet metabolic demand. It also increases survivorship of bleached corals. There was a natural bleaching event in 2014. We collected corals on the reef. Some were healthy and some were bleached like this, so we could collect pairs. And we put them in tanks for 8 months. Some as is, and some were fed. On the y-axis here is the number of corals that survived. So in the healthy corals, the gray bar, whether they were not fed or fed, these gray bars, they all survived the 8 months. But the bleached ones, 2/3 died if they were not fed, and more than 2/3 survived if they were fed. So feeding doubled survivorship of the bleached corals. So that's what we've covered here. These first two here enables bleached corals to meet metabolic demand, reduces mortality from heat stress. We also know from other studies we've done that feeding is essential. It's a source of nutrition for tissue building and lipid synthesis. We know that it minimizes heat stress impact and bleaching susceptibility, and we know that it's essential for long-term recovery. So we can detect their need for feeding and zooplankton to build lipids, to build fat for a year. They look recovered. They've been brown for a long time, and yet physiologically, they are still suffering or compensating for what happened a year before from bleaching. So there's this long tail of recovery from bleaching, and feeding is a really important part, not only to that recovery, but to their ability to sustain that bleaching in the first place. So we know feeding is important, and we also know that coral feed more when there's more to eat. So if we can get them to eat more, maybe it'll help them be more resilient and save more corals. So how can we get coral to eat more? Can we use technology? Turns out yes, because that's what I've been working on. I invented Uzella. It's the Underwater Zooplankton Enhancement Light Array. It's a fancy underwater flashlight. It is autonomous, programmable, deployable for up to 6 months on a single battery. It requires minimal maintenance, and it is provisionally patented. The way it works is zooplankton are attracted to light the same way that moths are attracted to a flame. So we program the device to turn on for an hour after sunset when it's dark and the zooplankton are migrating. And that's when coral like to feed mostly anyway. The zooplankton are migrating, the light turns on, attracts the zooplankton, and now concentrates zooplankton near the light. We put corals near the light, and now they have more feeding opportunities. And the idea is if they feed more, they'll grow more and die less. And it turns out that's exactly what happens. This is my team that's working on this. We're collaborating with folks in Hawaii. This is my grad student, Shannon, my postdoc, Anne-Marie, who just finished her PhD with me. This is our very first dive on our first trip when we did our first experiment using Uzella. We're in a really good mood. They weren't necessarily that happy by the end, but it's always good to start off in a good place. And we asked, do corals exposed to Uzella eat more zooplankton and grow more following heat stress bleaching? So do they grow more when they're heat stressed, but also when they're not heat stressed? We need to know both those things. And we did an experiment where we had corals, we put Uzella in a rack here. You can see it a little bit. Corals are all around it. Some are bleached. The white ones are bleached ones. The dark ones are healthy ones. And then we left them there for 10 days. And these, you can see the racks here. This one has Uzella in it. This one doesn't. We had controls. At night, the lights turn on, concentrate the zooplankton. And then we had controls, ones without the light, so we can see what the effect is of those two treatments. We want to know, does Uzella work better than just having them on that particular rack on that particular day? During those 10 days, we measured the zooplankton concentration. Is this working the way we thought it would? And whoops, I forgot. I've got a short video here to show you the experiment underwater. It's a little wobbly because you're scuba diving and holding the camera as you go. And then on the right is the lights at night, and you'll see that the little flecky stuff here, that's the zooplankton that are being attracted to the light, and then around it, the coral fingers are there. And so they're in the path of that light and where the zooplankton would be. Over those 10 days, we measured the zooplankton concentration. This is showing zooplankton concentration on the y-axis in number of plankton per meter cubed. And we measured different size classes. So these are all less than a millimeter, but coral really like the 150 to 400 micron size classes, just the right size. They'll eat all of them. That's their favorite. So we like to keep track of the different sizes and then the total. And in all cases, the number of zooplankton on the reef, which is natural, the green bar, and then on the blank, so that's the control right over the Eusella racks where there weren't lights, is the same. When we have Eusella, it's dramatically higher. So zooplankton concentrations are 4 to 7 times higher with Eusella. So we know Eusella is concentrating the zooplankton, and the corals have more opportunity to feed. After 10 days with Eusella, we allowed the coral to feed on that 10th day, and then we collected some of them, we killed them, we put them in the lab, and we dissected their guts, and we counted how many zooplankton they ate. Because the only way to know for sure if they're eating it is to actually dissect their guts. I will say that even though we do kill coral in our research, we don't kill the whole colony. We take a little piece, a branch, and because they're clonal, it just keeps growing. The parent colony on the reef is still alive and well. We only work with species that are very abundant at our sites, and we work completely permitted through the state of Hawaii. Okay, so we work again with Montipora capitata and Parietes compressa. On the left here is coral feeding, or the number of zooplankton they ate per 100 polyps. So we dissected 100 polyps, and we have to do that at night after they have fed, because even though we preserve them, zooplankton are really delicate, and they start to break down, and if we wait till the next morning, they're kind of mushy, and then we can't tell what it is. So we have to do it at night. We're up pretty late. There's often music involved. We are in a building that's air conditioned though, which is a plus, because the dorms are not. Okay, so we measured how much they ate, and in the Montipora capitatas and the Parietes compressa, whether they were bleached, which is the pink, light pink bar, or non-bleached, which is or healthy, which is the blue bar, when they don't have Eusella, they eat between 0 and 3 or 4 zooplankton per 100 polyps. This is very normal. We've seen this over and over again throughout all our studies. When they have Eusella, they eat substantially more. They eat 10 to 20 times more with Eusella. And this work is now in press. It just got accepted for publication. And we know from work that's in progress that they also grow about twice as much. This is work being done by my graduate student, Shannon. I'm not showing it because it's not published yet. But I will tell you that they grow about twice as much. We also think about baby corals, or recruits, because baby corals are a source of genetically unique combinations, and there may be some new unique combinations of individuals that are better equipped to cope with future ocean conditions, current and future ocean conditions. Unfortunately, about 95% of baby corals die in the first few months, and it's because they are like little gummy bears out there for fish, and they're yummy snacks, and predation is usually wipes them out. So the faster they get big enough that they're outside of that gummy bear phase, the better, because then they will not be susceptible to predation and they can grow. In addition, baby corals need a lot of zooplankton. They're not really good at photosynthesis yet, and so they depend more on zooplankton. And this on the right here is a single polyp coral. So when coral reproduce, they release either egg and sperm in the water column or bundles of egg and sperm in the water column. Larvae are formed in the water column. They swim around, and then they settle on a hard substrate and form a single polyp to start. And then that single polyp divides into 2, and then 4, and then 8, and it keeps growing and growing and eventually forms, in about 2 years, a colony about this big, depending on the species. Maybe about this big after about 2 years. And in about 4 years, it's about this big and reproductively viable. But they need food early. And so we thought maybe Eusella could help increase baby coral survival and growth. This is a super low tide. So this is actually the reef in Hawaii where we work, and it's exposed for a couple hours at these extreme low tides, these king tides, and then we of course had to do an OHIO for that. Yeah. We put some adult colonies, I know it's really dim here, in the tank. These are some adult colonies that spawned They're into the water column, they're larvae, and then we put these structures, these domes, in the tanks with them, allow the larvae to settle on the domes. We then put the domes on the reef, on the same racks that we had used in the previous experiment. And these domes are interesting because whoops, I didn't mean to do that. Well, have these holes in them, and that's so that the Uzella that we put underneath them, when it turned on, the light would shine through. And so you see the light shining, it looks like a disco ball, and the zooplankton come to it. And we left those out for 6 months. And we then went back and counted how many survived and how much they grew. And again, this is work in development by my graduate student, Shannon Dixon. And what we found is that the coral survivorship doubled and growth quadrupled. Right? So Uzella can really make a difference in coral growth, not just in adults and in feeding in adults, but clearly for the baby corals as well. So we think Uzella has a real role to play in coral restoration, in helping corals grow faster, so you can outplant them faster, in keeping colonies that are on the reef growing, surviving, less stressed, more resilient, and for implementing all kinds of potential functions for reef restoration conservation. So some examples of what we think Uzella could be used for are the military. This project was originally funded through a Department of Defense program, and it's the reef, or the R3D project in Hawaii, to build an artificial reef to where one doesn't exist, to protect coastline from sea level and erosion. My device is part of a strategy of technologies to help increase the resilience of the coral so that they can withstand heat stress and so that they can grow faster. The idea behind having a hybrid reef is you build the structure and then you glue corals onto it, and then the corals grow and it's self-sustaining. In addition, it is very difficult to build something out of concrete that does the job that coral can do for mitigating wave erosion. Because of the complexity of coral reefs and all those little nooks and crannies, it does a much better job than any physical concrete barrier. So the idea is here you start it with concrete and then you let nature take over and build it. But it only works if they live. And so my technology is part of that. You could also use it in existing restoration projects, something like this, where you might want to use Uzella to help those survive. You've already invested in this. You want to make sure they survive, they grow fast, that the new larvae that settle and reproduce have a better chance of survival. You might want to use it for different species. Like this is a red coral. It's used in the jewelry trade. It is harvested illegally for the most part. Coming up with a way to farm those and accelerating growth with the Uzella would be a way of lessening pressure on natural populations and putting it into a farming context. I have colleagues in Europe working on that particular problem. You could also use it for nurseries. Coral restoration is a two-step process. You take an adult colony, you take a little piece off. If you put it right on the reef right away, it's going to die. If it's too small, it dies. So instead, you either put it in a land-based tank system to let them grow till they're about this big, or you hang them in a system like this, these nursery trees. They're PVC trees with a weight on the bottom and a float on the top to keep them upright, and you hang your little piece of corals off of that and let them grow there. Once they're big enough, you then can put them out on the reef. Uzella could be really effective here because you could put Uzella right on the center of that tree. It can shine up through the tree, feed them more so that they grow more and die less. We are collaborating with the Coral Restoration Foundation in Florida to do just that now. And I've been funded through the Ohio Third Frontier program to commercialize Uzella. We're working with an engineering company, an Ohio-based engineering and manufacturing company, to redesign Uzella so that it's manufacturable and we can sell it so that it can help corals everywhere, not just in my little projects. In the work with the Coral Restoration Foundation, my team just got back yesterday. We are already working on that, and I'll tell you a bit more about it next. So this is what the nursery looks like here, these trees. Their site is several acres. It's the largest coral restoration nursery in the United States right now. We deployed last June Uzellas on a number of these trees like this. These are the acropora. These are the ones that keep dying in Florida. The acroporas, these branching ones, so we're working with them on ways to make them more resilient. We deployed Uzella on these trees. This is me. On the trees, and then we had trees without Uzella. My team just came back. We took samples at the beginning and pictures, and we took samples and pictures again last week, and now we can see how they did over that time, and I will report back if Uzella works as well in Florida as it does in Hawaii. This work is now being led by my postdoc, Anne-Marie Hulver. And then at night, this is just a pretty picture from the boat. You can see all the Uzellas that are on, and the team that we're working with there are fantastic. They're really committed to this idea. I presented this work in Mexico at the Reef Futures Conference, which is a lot of restoration, conservation-oriented folks, and there was huge interest. So we are now we're trying to set up a partnership with folks in the Red Sea at KAUST. They are on track to be the single largest restoration program in the world. And if our device works with them, then there would be a huge demand for it there as well. My goal is to save coral reefs. And if it helps coral reefs, then that's what we're going to do. So coral feeding is a key to coral survival this century. And we have shown that targeting good coral feeders and enhancing feeding opportunities are likely to increase coral restoration and conservation efficacy success. We're still early days, but everything so far is pointing to this. And Uzella is a technology intervention that can help corals survive this century. Is it going to help all corals everywhere all the time? No. Can it help some? Yes. And so we come back to our question, can we save coral reefs? We can. And there are multiple things that happen, and they have to happen all at the same time. But they all happen at different speeds. So we need to mitigate climate change and local stressors. That goes without saying. But that's a big global, that involves countries involved, that involves, right? That's way above my pay grade. But that needs to happen. And then in the meantime, we can do conservation and restoration, and to help that restoration and conservation be more successful, we can also integrate some technology. And Uzella is part of a suite of solutions that fits into this framework. So can we save coral reefs? Some of them, sometimes, and in some places. Thank you.
Okay, so we probably have some questions for Professor Grottoli. And as per usual Science Sunday's tradition, students get their first shot at the questions. Go ahead.
So if the corals rely on zooplankton, would since there's a lot of agricultural runoff in across the world, would it be possible to place artificial reefs at the end of river deltas and use that to kind of, I guess, fuel the coral growth, like create nurseries there?
Yeah, so yes and no. Yes and no. Great idea, though, and not one that hasn't escaped me. So a little bit of nutrient, which is what you get from runoff, does stimulate primary productivity, which stimulates zooplankton production, which is food for coral. And coral can do very well under those conditions. However, it's a fine line between a little nutrient and a lot, because a lot of nutrients completely collapse corals, and it really undermines them. They do really poorly at high nutrients. So it's a very delicate balance. But yes, in theory, controlled runoff with a little bit of nutrient maybe could be beneficial. The other problem is, though, if you have too much and there's too much particulate, it can smother coral. So smothering coral bad, too much nutrient decouples that symbiosis again. But a little nutrient goes a long way. So how to thread that needle is tricky. But I like how you're thinking.
Go ahead, young man. Just yell it out and then she'll repeat it.
I'll hear it.
So basically what I'm trying to ask is, like, Uzella in general, has it been, like, you've already told us how it's been a success in Hawaii and Florida, but theoretically in other environments that are, like, a little bit different, do you think it's the same technology and everything, but do you think it could work for, like, let's just say, the Australia or somewhere else too?
Yeah, so the question is, can Uzella work outside of Florida and Hawaii? And the basic biology of coral is the same everywhere. There are nuances between the species, like you saw when I showed 3 species, there are slight differences, but the basic biology is the same. So in principle, it should work everywhere, but we need to test that. We need to demonstrate that. And we are, once we get the device commercialized and we can sell it and we can more broadly disseminate it, then we can work with folks in Australia and Indonesia and all over and test it in multiple scenarios. Yeah.
All right, go ahead, Miss.
I was wondering, with that Uzella, you were saying that basically everything would die around 2050, but with Uzella, would it slow down the processing of death, or like at least keep everything from going into extinction?
So will Uzella prevent corals from going extinct? Maybe. I don't know how many, maybe 1 million Uzellas, 1 billion Uzellas, I don't know. I don't know. Right now we're trying to prevent definitely corals from going extinct and keeping some reefs functioning through conservation, restoration, and technology. Whether that's enough to prevent extinction by 2050, I don't know. It depends on how much we mitigate climate, or if we accelerate warming more. If we accelerate it more, we're on track for that too. Maybe we buy a couple decades, but it's futile. Maybe corals adapt in ways we hadn't anticipated, especially when we start working with baby coral, and Uzella are even more effective than we originally thought. And so we have pockets of preserved reef that are highly functional that'll get us to the end of this century. But I think there are way more questions than answers there. Yeah.
Okay, sir, in the back, go ahead.
So obviously Uzella looks like it has a lot of promise in helping coral to thrive better. Are there concerns about light pollution associated with it and how it affects organisms that rely on light cues for vertical migration in the water column, or larger animals that are using light cues for nesting and things like that?
For sure. So ambient light at night is a real thing. We know that there are negative effects of ambient light at night when you have chronic, persistent light shining down. So think a roadway beside the ocean and the street lights are on all the time, or a pier that's sticking out with the light shining down all the time, boat traffic light shining, or not boat traffic, but like a pier, like fixed structures where the lights are shining down all the time. There are known negative effects on some organisms. Coral, a little bit, not so much. And so, yeah, there are some costs. In the case of Uzella, though, it shines up, not down. And it's very dark around the coral and it's only on for 1 hour. And you wouldn't necessarily need to use it all year. Coral, when they reproduce, the way they time the release of their egg and sperm, it's coordinated with the lunar cycle. And there was some concern that it would interfere with their ability to detect the lunar cycle and interfere with reproduction. But a colleague of mine who works on reproduction in corals right beside where we were doing our Uzella tests said they spawned just fine the year we had Uzella out. So maybe they were used to it and it was fine. I think because it doesn't shine down, it's not the same. However, there may be costs we have not anticipated. There may be risks we have not anticipated. And so one of the things I think about is if you have that decimated reef, like those pictures I showed at the beginning where it's rubble, and you put Uzella out there, that's any cost associated with that is going to be way less than the cost of not having a reef there at all. And so in the cost balance, I think in extreme conditions where we are doing restoration, that's where Uzella is going to be targeting. We have more to gain than to lose.
Go ahead, sir.
I have 2 questions for you. One is, why does the coral expel the algae when it gets too warm? And then so it bleaches. And the other is, has any thought been given? I'm sure it has been given. What is the thinking around stocking zooplankton? In other words, breeding, having tanks where you breed them and
Yeah.
let them go over reefs.
Okay, so stocking zooplankton requires a lot of manpower, energy, resources, and you got to do it every day. And then it all gets dissipated when you put it in the water. Whereas Uzella is leveraging the natural zooplankton abundance that's already there. It's just focusing it a little bit, and it's not even affecting the concentration anywhere else. There's a lot, but it's bringing it to the coral in a focused way. The algae leave coral when they're bleached because of reactive oxygen species production by the algae becomes toxic for the host, and they kick them out. That is the leading theory. There's evidence to support that, but that's what we think is happening.
Front row.
Did you have an aha moment? And then how long has it been since that moment for you to develop this technology? And what does the commercialization process look like for you guys?
I've been thinking about Uzella for a while. The aha moment for me came not with Uzella, but when I was a graduate student at the end of my first year, I did a field course for graduate students in Hawaii on coral bleaching. And I was running an experiment in tanks, and I'm in my bathing suit, cleaning the tank, and I kind of sat up and I went like, well, I could do this for a living. That was my first aha moment. And then I completely, I rejigged everything, and that's what I ended up doing. Then with Uzella, I've been thinking about versions of Uzella. I actually used lights to feed corals when I was a grad student. It didn't work the way I thought. There was a lot of learning that went on, but I've been thinking about coral feeding since I was a grad student. And when I was a graduate student, when I first proposed feeding my corals, I was testing light and feeding. And when I first said, well, I'm going to feed them as well as evaluate light, because those are the two parts of the carbon equation. I was thinking about carbon in corals. And one of my advisors thought that was hilarious, because we know that coral can meet 100% of metabolic demand from photosynthesis. So it was always thought that feeding was sort of ancillary, like this cute thing they did on the side that meant like some little bit of nutrients here and there but that was like 1994, dating myself, 1994. And at the time, there just wasn't an appreciation for the role of feeding, even though corals are animals with mouths and tentacles. They're related to jellyfish. I don't know if that fully answers. Oh, the commercialization. We are very early stage, so we have the provisional patent, the phase 2 of the provisional patent, and we have the grant to develop a manufacturable version. So what we made is a homemade thing that costs a lot of money, because my grad students literally, soldering the board and doing the programming. This is not scalable, but the company we're working with, we saw some designs last week that look amazing. It's smaller. It's going to have software to support it. So right now we're just hard coding it every time, but this will have a software interface, which you need for users to be able to use. It'll work on a rechargeable battery, so it'll be more environmentally friendly. Yeah, so we're working on that. Thanks.
Miss, go ahead.
What would happen if the coral overate the zooplankton?
No, that's a really good question. What happens if corals overeat? I actually inadvertently tested that one time. When I was a graduate student, my first experiment, I had 4 levels of feeding and 4 levels of light that I exposed my corals to, and the ones where I let them eat 100%, so every mouth was full every other day, so they were just stuffed. It actually started to decouple the symbiosis a little bit. They looked great. They looked fantastic, but algae have to share their sugar with the host because they still get stuff from the host. But when I overfed the coral, they were getting so much nutrient, they didn't need the host as much, and so growth slowed down. But it was at the extreme. In nature, they would never be able to eat that much. It's not concentrated enough. But in theory, yes, it does. Too much is no good either. Yeah.
Let's see, front row.
So I was really struck at the beginning of your presentation when you talked about how 90% of Florida's coral has disappeared since 1970, which immediately got me thinking all through the presentation about the scalability of being such a massive problem. And you also talked about the economics of impact of coral. So can you talk about the economics of actual restoration? Think about how much intensity of resources did it take to save a certain area of coral? So I can start to think about sort of the cost.
Yeah. So way outside of my wheelhouse, but I will, he's asking about the cost of restoration, the economy of scale in restoration. And we are so at the beginning of restoration going to scale. So the Australians are investing 10 million a year in all kinds of crazy technology. Not crazy, but like they're really going outside the box and thinking of different technologies they can use so they can scale up restoration. And that's testing it. Florida is spending Mission Iconic Reefs with NOAA. I don't remember the number. It is millions a year. But again, still very much in the testing phase, still very much in figuring out what works best, how to scale up, because what works in Florida is not going to work in Australia. What works in Australia is not going to work in the Red Sea. Every place is different. Every ecosystem has their own nuances. But I think that is still, like, it is expensive right now. It is expensive. That's why it's not being done everywhere all the time. It is being done in focused areas. So there's a 30 by 30 mission, and that is to protect 30 reefs or like a 30 by 30 kilometer area in 30 places in the world or something like that. That's like this UN level goal. Who pays for it? Who does it? That's all in the details. There is a big program coming out of the G7 that is being led by Saudi Arabia, because they were head of the G7 at the time that it was established. They are funding $10 million a year to 10 different projects, so $1 million each, for restoration and restoration technology. And all of these programs are thinking about what are the right combinations of things so we can scale up, so we can maximize how much reef we can save. But we're not at the point where we can say, this is how we do it, this is how much it costs, you have 10 hectares, it'll cost this much. We're not there yet. But we're working really, really hard to get there.
Thank you.
Yeah.
Okay, let's take two more short questions. Go ahead.
Okay, thank you. Obviously, there's multiple solutions that are needed for anything. It seems like there's never going to be a silver bullet. I remember reading about the zooxanthellae trying to look at different, gene splicing or DNA or like doing these super zooxanthellae. And I don't know if your lab works with that as an additional treatment to try and find out. I was very interested to see that some corals only like once.
Yeah.
Much more difficult. But is there a zooxanthellae that's kind of ubiquitous in all corals, or do you see promise on that?
Yeah. So So the question is, the endosymbiotic algae, can you genetically manipulate them, or can you force coral to take up one species that's more thermally tolerant or more resilient than another? And the answer is sort of kind of no. Sometimes yes, sometimes no. Some coral will take up other endosymbionts, but there's a cost. So thermally tolerant endosymbionts are good at being thermally tolerant. They're not always the best symbiont partner. They're not always the best at sharing. So we often see decreased calcification in corals that have thermally tolerant endosymbionts, which is better than dying. You just grow less. And so some people are saying, well, we can do that. Forcing coral to take them up can be done in a lab. Doing it at scale, much harder. Genetically modifying organisms, there's a whole suite of people trying to use CRISPR and other techniques for inserting genes, but we don't even know what gene to target in the first place. So there are certain heat shock proteins that are helpful. Are we going to get the right one? It's not like medicine, where there's a gajillion dollars in a gajillion labs simultaneously working on these problems, and you can do an experiment on a benchtop in 2 weeks and have an answer and then do the next thing. We have a fraction of the resources, a fraction of the researchers, and we have to do it underwater. So it adds a layer of complexity, but those are the right questions. Some people are doing breeding. So you're taking coral that survived a bleaching event with another one, and you're cross-fertilizing those to produce offspring that are, when you test them, are more thermally tolerant, and so are their F2s. Their next generation are more thermally tolerant. Does that mean they're good at fighting disease also? Maybe not. There are usually trade-offs. So these are all things that are being investigated. and trying to find like the optimal solution between cost, time, investment, effect is really like it's an all hands on deck thing. Yeah, thanks.
Okay, last question in the front row here. Go ahead.
Is the zooplankton highly resistant to ocean temperature increases?
No. All marine organisms are sensitive to swings in temperature, either too low or too high. That's true of all organisms. We don't like it to be too hot or too cold either. People die when it's too hot, people die when it's too cold. Same for all organisms on the planet. Zooplankton are no exception. Not too many people study zooplankton, especially in coral reefs. And there's some evidence from open ocean data that zooplankton in the open ocean, that their abundance has been declining over time, and that's associated with climate change. So there's a risk that there will be fewer zooplankton on coral reefs right when corals need them most. But maybe we go from 10 to the 3, like 5 times 10 to the 3 to 4 times 10 to the 3 per meter cubed. I don't know. That's probably still enough for coral, but I don't know. They're also susceptible to ocean acidification because they have kind of shell things. So are coral, but right now that's not their problem. Their problem is heat. 20 years from now, the acidity will become a problem too.
May I make a very quick comment?
Yeah, go ahead.
Yeah, I just want to let you know that this was an outstanding presentation.
Thank you. Thank you.
So in academia, we have a thing where people say, this is really more of a comment than a question. And usually it's not very nice. That one was nice.
Yeah.
So today, for the reception, you'll have a chance to ask Professor Grottoli some more questions. Now, how do you get to the reception? Don't go to the usual place. Instead, basically cut across the lobby diagonally to what's called the Great Hall. So it's forward and left. And an hour or so ago there was a bunch of like video game dudes in there, but there's like 300 of us, so I think we could take them. And should be all right. Let's thank Professor Grottoli.